Hello students, today we are going to discuss about radiation sterilization. This is our main topic, that is radiation sterilization. First we are going to discuss what basically the sterilization is. The sterilization is the process by which we are killing the bacteria, whether that bacteria is in vegetative form or that bacteria is in endospore form. Now we are having different techniques to carry out the process of sterilization, like wet sterilization is there, like dry sterilization there, and similarly we are having a type of sterilization that is called as radiation sterilization. Now what is this radiation sterilization? It is the process of sterilization in which radiations are used to kill the microorganism, whether that microorganism is bacteria, virus, fungi, spores on any surface or any objects. These microorganisms will be killed by the radiations. Now these radiations which we are using for the purpose of sterilization are being used in the medical field, pharmaceutical industries, as well as the food industries in order to sterilize the medical equipments, right? And second one is drugs, and as well as in the food industries for the sterility. Now two types of radiations are mostly used, that is gamma rays and ultraviolet rays. This gamma rays, it's an ionizing radiation, and the ultraviolet rays, these are the non-ionizing radiation. Ionizing and non-ionizing. Gamma is ionizing radiation and ultraviolet rays are non-ionizing radiations. Now first we are going to discuss about the gamma radiations. Basically these gamma radiations, they consist of the high energy photons. These photons are basically the packets of the electromagnetic energy, and these gamma radiations, they destroy the microorganism by breaking their molecular bonds which are there in the bacterial or microbial DNA, right? Now when the microbial DNA is distorted or when the microbial DNA is damaged, the microorganism is non-viable or the microorganism is dead. In simple or in nutshell, we can say these gamma radiations, they destroy the microbial DNA, and when the microbial DNA and other cellular structures are damaged, right, the bacteria is dead, or we can say the bacteria is non-viable. Now we are having certain sources of the gamma radiations. First is cobalt 60 and second one is cesium 137. These are the radioactive isotopes basically which emit these radiations. These are radioactive isotopes, this cobalt 60 and cesium 137. They'll continuously emit the gamma radiations, and these gamma radiations will be used for the purpose of sterilization. Now as discussed earlier, that these gamma radiations, they'll particularly attack the bacterial DNA, right, and they'll cause the ionization of the bacterial DNA, and when the DNA of the bacteria is distorted or when the bacterial DNA is damaged, the bacteria is non-viable. Now these gamma radiations, they are having the capability or they are having the ability to penetrate deeper into the material which is being sterilized. Suppose if we are sterilizing an article which is densely packed with the linens or with the clothing which is being used for the purpose of sterilization, it is having the capability that it can penetrate deeper to the sterilizing article or to the sterilizing material, right? Now these high energy photons, they'll come in contact with the biological molecules. We are talking about the microorganisms. These biological molecules, these are referring to the microorganisms, right? And they attack the DNA of the bacteria and will cause ionization, and that ionization will disrupt or that will damage the DNA strands of the bacteria, leading to lethal mutations of the bacteria. That means it will mutate the bacterial DNA, and when the mutation will occur, the bacteria is dead. It also affects the enzymes as well as the proteins. When it affects when it damages the bacterial enzymes and proteins, the bacteria is non-viable. Now applications, certain applications over which or at which we are using these gamma radiations for the purpose of sterilization. In the medical field, they are used for the purpose of sterilization, especially for syringes, for catheters, and for the sutures. These radiations are used in the medical field for the sterilization of syringes, catheters, as well as for the sutures. And as discussed earlier, that they are used for the sterilization of the pharmaceutical industries, for the drug packaging, and as well as for the pharmaceuticals or for the drugs which are not able to tolerate the heat, like we are using heat in dry or in wet sterilization. Over there we are using the temperatures of 121 degree centigrade, 134 degree centigrade, or 160 to 180 degree centigrade. But over here we are not using any kind of temperature, only we are using the radiation. That radiation is gamma radiation. And apart from pharmaceuticals and medical devices, this is also used for, this gamma radiation is also used for the sterilization of the tissue grafts, bones, as well as other biologics. These gamma radiations are used to eliminate the contamination. Now there are certain advantages of this gamma radiation. First is deep penetration. Now as discussed earlier in the slides, that these gamma radiations, they are having the capability that they can penetrate deeper. If an object or if an article or if an equipment or if an instrument is densely packed with this sterilizing material, right, or with the material which is being used or which is being used for the packing purposes, right, it is having the capability that it can penetrate deeper, and upon penetrating deep, it will come in contact with the equipment surface or instrument surface, and over there the microorganisms, right, they will be killed. And second one is sterilization of heat sensitive products. Now, as discussed, that in the gamma radiations we are not using any kind of heat like in dry sterilization or in wet sterilization, right? Now the equipments or the instruments, suppose if the instrument is made up of plastic, or we are using it for sterilizing a biological tissue, or the medications which are not able to tolerate the heat, right, this method of sterilization will be feasible for the plastics as well as for the biological tissues as well as for the medications. Since heat is not being used in the gamma radiation sterilization, there are no chances that it will disrupt or it will destroy the plastics or biological tissues or medications. Now this is also having the high efficiency that it can damage the wide range of microorganism, whether that microorganism is bacteria, virus, or endospore. Now there are certain disadvantages also. Apart from advantages, there are certain disadvantages. First is continuous radiation. Now as we know that we are having a source of gamma radiation is cobalt 60, right, and cesium-137. They continuously emit the radiations. This cobalt 60 and cesium-137, they'll continuously emit the radiations, gamma radiations, right? And at that time we may require some protective measures in order to protect the workers or the individuals who are dealing with the gamma radiations as well as the environment. And second one is degradation of material. Now if the object or if an article is prolongedly exposed to the gamma radiations, right, it can damage them, right? Suppose if a polymer means the material which is made up of different kind of molecules, right? Now what this gamma radiation will do, it will break the molecular bonds in the polymer and it will destroy it, right? And third one is high cost. When we are using the isotopes or sources of these gamma radiations, they are costly, as well as the safety measures which are being followed for in order to protect our environment or in order to protect the workers, that is also costly.